All right, class, so welcome to Wednesday, November 3rd. We're already in November. I can't believe it. Today we'll be going over chapter 14, the lymphatic system, as well as your immune system. We kind of put those two systems together. You will have a lab activity if you're in Wednesday's lab today. And then next week, the important thing to note is that you will have your lab practical 3, and it's on exercises 10, 11, 13, and 14. And if you're wondering what those exercises are, the easiest way is to go to either your downloadable PDF lab manual from the syllabus, or go to this virtual lab link. And if you guys don't have this link, let me just go ahead and put this in the chat. I sometimes wonder if people use this. I think you guys do. But if you enter into our lab, you can see what exercises. So we have 10, 11, 13, and 14. So your lab exam next week will be on the nervous system, special senses, blood, and then the heart. So that's your lab exam 10, 11, 13, and 14 for next week. So just keep that in mind. You have a lab exam next week in lab. Also, if you're listening to the recording and you're not here, what I did for your lecture exam is I added 2 extra points to everyone's score, whether you got it right or wrong, to make up for a question that was about clotting factors that we didn't go over. I'm not even sure if it's really in your textbook. I think I told you to study it to try to help you out to see if anyone could figure it out, but still we didn't really go over that one. So everyone kind of got 2 extra points on the lecture exam. And if you ever want to go over a lecture exam, come see me in my office hours, Thursdays at 11:30, or stay after lab one day, or we can set up another time to go over that. So here we are at chapter 14. The lymphatic system is the official body system name of the system we're going to study, but we sometimes, or you might hear it referred to as the immune system as well. So I like how this chapter labels it. It's the lymphatic system and immunity, because we don't really have an immune system. The official body system name is your lymphatic system, and then immunity is a part of that system. So the functions of your lymphatic system, no one really knows a lot about this system. It helps a lot with fluid balance, fat absorption, and defense, and that's where our immunity part comes in. Basically, the lymphatic system is kind of your body's catch-all drainage system for extra fluid that is in your tissues. So if you think of your blood as the way, in your cardiovascular system, your blood transports nutrients, gets rid of wastes. Well, there's other fluid that can build up in your body's tissues. And that kind of excess fluid that can't be transferred or transported in the blood gets transported through your lymphatic system. And this shows you the lymphatic system, the different lymphatic ducts, where they're located. So basically everything in green is a lymphatic duct. You have lymphatic nodes or lymph nodes. All of this kind of works to drain excess fluid in the body. It takes it to the spleen for processing. Your lymphatic system also has to do with immunity that we'll talk about. But this is kind of your body's drainage system for any excess fluid that your blood can't transport. Your lymph nodes, and a lot of times they're found kind of in the cervical region in the neck, can get inflamed if there's an infection in the body, because there's more blood cells, right? White blood cells circulating. So that kind of swollen lymph nodes is a sign of infection, because usually that means there's more drainage going on, or more white blood cells building up in the body. So components of the lymphatic system, we have lymph, which is the actual fluid that enters the lymphatic capillaries and those ducts composed of water, some solutes. We have lymphocytes, which are cells, lymphatic vessels, lymph nodes, and then your tonsils, your spleen, and your thymus gland. So those are all components of the lymphatic system. The lymphatic capillaries carry fluid in one direction from the tissues to the circulatory system. So your lymphatic capillaries might drain excess fluid from your tissues and then bring them back to the blood, your circulatory system. Fluid will move from blood capillaries into the tissue spaces. The lymphatic vessels are tiny closed-ended vessels, so they end at a certain point. Fluid moves easily into them. They're located throughout most tissues, and they will join to form the lymphatic vessels. Lymphatic vessels are like small veins. The lymphatic capillaries join together to form these vessels. They have one-way valves in them to just ensure that the direction of the lymph flow is going in one direction. The right lymphatic duct is where all the lymphatic vessels from the right upper limb and the right head and neck and chest region empty. And this right lymphatic duct will empty directly into the right subclavian vein to go back into the heart eventually. The thoracic duct is where the rest of the body empties from the lymphatic vessels. It empties into the left subclavian vein. And this is just a look about how lymph is kind of formed and then the movement of lymph. So we see here, in green, we have the lymphatic vessel. You can see how fluid will move either from the arterial into the tissues and then into the lymphatic capillaries. We have tissue cells. So kind of the lymphatic vessels are right in there with your arteries and veins and tissues to again help drain excess fluid from your tissue cells in the body. This picture is just showing the one-way direction of the lymph flow with the valves opening and closing, similar to the valves that we had in our veins to help the unidirectional flow back up to the heart. Then our lymphatic organs, we have the tonsils, the palatine tonsils, the pharyngeal tonsils near the opening of the nasal cavity, and then the lingual tonsils on the posterior surface of the tongue. And your tonsils just form kind of a protective tissue or ring of lymphatic tissue around the nasal and oral cavities. This is probably a good place to have a protective ring of lymphatic tissue because you have a lot of things entering the body. All the air you breathe, anything you put in your mouth is entering. So this is kind of just an extra defense mechanism that these tonsils make up of lymphatic tissue to kind of help act as a barrier for everything that enters the nasal cavity and the mouth. So here we kind of see where these tonsils are located: pharyngeal tonsils, palatine tonsils, lingual tonsils. Again, these tonsils can get inflamed within an infection, and that's what doctors will look for if they're looking into your mouth. You might need your tonsils removed at some point. So those are the tonsils, forming a ring of protective lymphatic tissue. The lymph nodes are rounded structures that vary in size. They're located near lymphatic vessels. They're in the groin, the armpit, and the neck. Lymph will pass through the lymph nodes before entering the blood. Lymph moves through and into kind of the immune system, and in the lymph nodes, it will be activated, and this is where lymphocytes will be produced if anything foreign is detected. And in the lymph nodes, you'll have the removal of anything foreign, any microbes, by macrophages. So again, this is why your lymph nodes might enlarge and become swollen, because this is where you'll have lymphocytes produced. You'll have microbes trying to macrophages trying to get rid of any microbes or anything foreign. So because you have extra activity going on in your lymph nodes, in the case of an infection, they will get bigger. And again, doctors will often feel your neck if you come in and complain of being sick. So this is what a lymph node looks like. You see different lymphatic vessels kind of bringing lymph into the lymph node and then bringing it out. It will have a blood supply to it. I don't think necessarily you need to know the specific structure of a lymph node, but this is what it looks like. The spleen is a part of one of your lymphatic system organs. It's the size of about a clenched fist, and it's located in your left abdominal quadrant, kind of to the left of your spleen, right behind, or the left or behind your pancreas. And the spleen will filter blood and detect and respond to anything foreign. So it will destroy old red blood cells. So this is where your red blood cells go. After about 120 days, your red blood cells need to be recycled. They go to the spleen, where the spleen breaks them up, kind of recycles red blood cells. And this is also known as kind of one of our body's blood reservoirs. So you'll have kind of a reservoir of blood hanging out in the spleen to help filter it. The spleen is made up of white pulp and red pulp. The white pulp is the lymphatic tissue surrounding your arteries, and the red pulp will contain macrophages and red blood cells that connect to veins. So again, our spleen contains macrophages, which again is a type of white blood cell of your body's defense mechanisms that will eat up or engulf anything foreign. So your spleen is also an organ that can enlarge in cases of infection. Specifically, I'm thinking of mononucleosis. Your lymph nodes can swell. The spleen can be painful because they're trying to fight off this infection. So this is where your spleen is located. It will have a blood supply to it. You can see the white pulp, and then the red pulp is surrounding the white pulp. The thymus gland is a bilobed gland. It's located in the mediastinum, which is the area between the pleural cavities. So it's behind the sternum. If you feel your sternum, the mediastinum is right behind that. The thymus gland stops growing at age 1, and then at age 60, it starts to decrease in size. And the thymus gland is what produces and matures the lymphocytes. And a lymphocyte is a type of white blood cell that we'll talk about. So here we have the thymus gland. It's located in the mediastinum region. So again, kind of the area between the pleural cavities, right above the heart. It'll be kind of in the area where your aorta is, the trachea. So here's your thymus gland. You'll see lymph nodes kind of attached to it. So that's your thymus gland, helps to produce lymphocytes. So here's kind of a nice overview of the lymphatic system. If you're still kind of confused at why we have this system and what it's important for. Again, we're kind of going to go through in detail how some of these cells are formed, because the cells that are formed in the lymphatic system will go on to help to form our immune system. But just in general, you know, we have arterial and venous circulation, and then excess fluid tissue will be picked up by these lymphatic capillaries. It'll be brought to the lymph nodes where it's filtered out through more lymphatic capillaries, and then usually back into circulation once that lymphatic fluid has been filtered. Also in the lymphatic system, we have lacteals, which help to absorb lipids and fats in your digestive tract, and that also enters the lymphatic system. The spleen is a part of that, and then we'll get into our bone marrow, thymus, other lymphatic tissue to see how different lymphocytes, which are B cells and T cells, which will go on to form a part of our immune system or our body's defense mechanism. So now we'll talk about immunity, or the immune system, which we again combine with your lymphatic system, because the lymphocytes that are produced in your lymphatic system will go on to help with immunity. What is immunity? What is the immune system? Well, immunity is the ability to resist damage from anything foreign that your body takes up. So these can be microbes, toxins, cancer cells. Immunity can protect against that. We have two different types of immunity, innate and adaptive, and we'll go over those two types. Innate immunity is present at birth. This will be a defense against any pathogen. It's accomplished by a physical barrier like your skin, chemical mediators, other cells, and an inflammatory response. For example, swelling, red blood swelling, hematomas form. So this is innate immunity that you are born with. This is present at birth. Physical barriers is the first line of defense. This is your skin, its mucous membranes, such as the mucus inside your nose, the mucus inside your trachea, that again tries to trap anything harmful that you breathe in. Tears, saliva, and urine can all wash away pathogens, which are a part of the physical barrier. Chemical mediators are chemicals that can kill microbes and prevent their entry into cells. So we have lysozyme, which is an enzyme found in your tears and saliva to kill bacteria. And then the mucous membranes prevent the entry of microbes. And again, mucous membranes are found in your nose, your trachea, anything to prevent anything foreign from entering your body. Histamine is a type of chemical that promotes inflammation by causing vasodilation. So it will dilate your blood vessels to try to bring excess blood cells to the source of infection to fight it off. Interferons are proteins that protect against any viral infection by stimulating the surrounding cells to produce an antiviral protein. Then we have the cells of the immune system. So your white blood cells are produced in red bone marrow and the lymphatic tissue that fights foreign substances. White blood cells are also known as leukocytes. We have 5 different types of leukocytes that we'll go over. Phagocytic cells are any sort of cell that phagocytizes something. And again, phagocytosis is eating something. So these cells will ingest and destroy foreign substances. And the two kind of big examples of white blood cells that are phagocytic cells are neutrophils and macrophages. Then we are going over, these are some of the types of the white blood cells. Neutrophils are always the cells that are first to respond to the infection, but they'll die quickly. Eosinophils are produced in red bone marrow to release chemicals to reduce inflammation. Basophils are made in red bone marrow, and they will leave the blood and actually enter the infected tissues to release that chemical histamine. Macrophages initially were monocytes, which is a type of leukocyte. They will also leave the blood, enter tissues. Macrophages are kind of our big phagocytic cells, so they can ingest even more than the neutrophils. They're just big cells that can eat up and get rid of anything foreign. They will protect the lymph in the lymph nodes in the blood. They're found in your spleen, your liver, and they're given specific names for where they're located throughout the body. So for example, macrophages, if they're in the liver, they're called Kupffer cells. Mast cells are made in the red bone marrow. They're found in your skin, lungs, the GI tract, the urogenital tract, which release leukotrienes, which help to fight off infection. Natural killer cells are a type of lymphocyte. They're produced in the red bone marrow as well. They will recognize classes such as tumor cells or a virus-infected cell, and they'll release chemicals to lyse the cells. So what natural killer cells will do is they will kill cells. They will go to the cell and they'll release a chemical that will cause the cell to lyse. Lyse means split apart. And when that cell lyses or splits apart, it kills it. So that's a natural killer cell. So what is the inflammatory response? When you get an injury, if you have anything foreign, the inflammatory response will involve chemicals and cells due to injury. It will be signaled by the presence of the foreign substance, and it will stimulate the release of some of these chemical mediators to start fighting off this infection. So let's go through kind of what happens during the inflammatory response. Let's say anytime bacteria enters the tissue. So maybe bacteria will enter if you get a splinter or you're stung by, stung by an insect, or you just cut yourself with a knife and the piece of that knife has some bacteria on it. So that bacteria enters the tissue, it will damage the tissue, chemical mediators will be released, chemotaxis, increased vascular permeability will occur, meaning that blood flow will be diverted to that region to try to increase blood flow to that region, because you might be losing blood. There will be an increased number of white blood cells, which are your body's kind of fighting cells against infection. There will be chemical mediators released at the site of the tissue damage. Bacteria will then be contained, destroyed, and phagocytized, meaning they'll be eaten and destroyed. The bacteria will be gone and the tissue will be repaired. Or if the bacteria remain, additional chemical mediators will be activated. And this makes sense. If you do get a piece of wood, let's say stuck in your finger, remember I got a big piece of wood in my toe when I was in college, and I eventually had to get it removed, but you know the bacteria is gone when the tissue completely repairs itself, the color of the skin goes back to normal. If the color of the skin remains red, swollen, you might notice red or lines moving up your leg, which means the infection is still there and your body is not fighting it off. So you can usually tell if the infection is gone or it's remained. Then adaptive immunity is defense that involves specific recognition to a specific antigen. And again, an antigen is a protein on a cell that can cause recognition of that cell. This immunity, adaptive immunity, is acquired after birth. So this is something that you get after birth. It reacts when an innate or an inborn defense doesn't work. It's much slower than innate immunity, and it has memory. And it uses lymphocytes, B and T cells, to remember kind of specific antigens or foreign substances in the body. There's two types of adaptive immunity. They can be antibody-mediated. So your antibodies can recognize a foreign antigen and then remember it. or cell-mediated. So an antigen is a substance that can stimulate an immune response. An antigen can be a bacteria, a virus, a pollen, a food, a drug. A self-antigen is a molecule produced by the person's body that stimulates an immune system response. And then an antibody are proteins the body produces in response to an antigen. So we talked a little bit about this when we were talking about blood. You know, you have antigens on your red blood cells, and then you have antibodies that fight off opposite antigens. And now we're talking about the terms kind of in a more general sense when we're talking about adaptive immunity and how your body can remember when something foreign enters it. The origin and development of lymphocytes. So we'll talk about adaptive immunity and how your body can remember and fight off infection. But first we need to talk about what a lymphocyte is that gives the body the ability to do that. So a stem cell is found in the red bone marrow. This is what that hematocytoblast was, the stem cell of your red blood cells that will go on and differentiate to all other blood cell types. So you have one kind of type of blood cell from your red bone marrow, and it will go on to form red blood cells, white blood cells, or lymphocytes. And lymphocytes are a type of white blood cell that will give rise to pre-T cells and pre-B cells. Lymphocytes are involved in this adaptive immunity that you acquire after birth. They develop from stem cells and they will differentiate or kind of change into their specific lymphocytes like B and T cells. So we're talking about kind of lymphocytes here. It's a class of our white blood cells. And now we're going to talk about two types, the B cells and the T cells. So B cells are a type of lymphocytes. They're involved in antibody-mediated immunity. So the ability for your body to create antibodies to remember anything foreign that would enter. They originate from stem cells. They will mature in your red bone marrow, and then they'll move to the lymphatic tissue after they're mature and they're ready to go. The B cells, what you guys should remember, you know, there's a lot of information being thrown at you in this chapter. The B cells produce antibodies. So your B cells, type of lymphocyte, produces antibodies. The T cells are also a type of lymphocyte. They're involved in cell-mediated immunity as well as antibody-mediated immunity. They will mature in your thymus gland. They'll move to the lymphatic tissue after they mature, and you have 4 different types of T cells. So we kind of started with lymphocytes, then we broke those down into B and T cells. Now we're breaking down T cells into 4 different types. So we got a lot of information here. So this is just showing the origin and processing of the cells. We have the stem cell, which differentiates into a pre-T cell and pre-B cell. The B cell matures in your red bone marrow, but the T cell matures in your thymus, and then they go on into circulation into the lymphatic system, because the lymphatic system is what will carry these B cells and T cells around through the lymph nodes and lymph vessels. So you're starting to see why lymphatic system and immune system are the naming is very closely related. The antigen recognition, so lymphocytes, which are cells, the lymphocytes have an antigen receptor on their surface. They are called B cell receptors if they're on the B cell, and the T cell receptors on T cells. And these receptors only bind with a specific antigen. And when an antigen receptor combines with that antigen, the lymphocyte will be activated. And this idea of adaptive immunity begins, or this idea of remembering, recognizing a specific antigen that's foreign and being able to always remember what that is. The MHC complex, major histocompatibility complex molecule, is the MHC is what we call the kind of the complex that contains the binding sites for these antigens. So it's a complex on these cells. It contains binding sites for antigens. It's very specific for certain antigens. It can hold and present a processed antigen on the surface of the cell membrane. It can bind to an antigen receptor on B or T cells and then stimulate some sort of response. So I'll show you how everything kind of interacts here in a little bit. Cytokines are proteins secreted by a cell that will regulate neighboring cells. So for example, interleukin-1 is released by macrophages that helps to stimulate helper T cells. So these are cytokines that are involved in the immune response. They're proteins that are secreted by a cell, but they help to regulate our surrounding cells, basically to kind of say, hey, we need more help. We need more helper T cells. Those are what cytokines are. So this is the proliferation of helper T cells, or how helper T cells are divided so we can make more of them. Here we have an antigen, which is again anything foreign. The antigen will kind of be connected to or bound to the MHC, that kind of class molecule. When the antigen is bound to that receptor complex, this will present the processed antigen to the T cell receptor of the helper T cell. So this MHC complex is just kind of a way to kind of grab hold of that antigen and then process it so the helper T cell understands what's happening. So this is our helper T cell. Interleukin, which we talked about, interleukin will stimulate the helper T cells to secrete interleukin-2 to produce interleukin-2 receptors. And again, interleukin-2, these interleukins are proteins that tell neighboring cells to help stimulate the production of more helper T cells. And so we get more helper T cells to divide to create more because we need more helper T cells to help and to be activated to help with this immune response. Lymphocyte proliferation. So the idea of proliferation just means the idea of kind of creating more lymphocytes. After an antigen is processed and is presented to a helper T cell, the helper T cell produces these interleukins and interleukin receptors. Interleukins will bind to the receptors and stimulate more helper T cell production. Helper T cells are actually needed to produce B cells, and B cells are what will produce your antibodies, which will eventually then remember the type of antigen that was initially kind of processed and be able to fight off against that antigen. So this is the proliferation of B cells. Again, we have what we call an unprocessed antigen. That means your body doesn't really know what it is yet, but it's something foreign. It will connect with a B cell receptor, go through our MHC molecule, that kind of way to kind of that processes the antigen that tells the helper T cell what it is. That helper T cell then releases interleukins, which helps then the interleukins are kind of like our, we need more help guys, the proteins. So they'll tell B cells to divide more to create more B cells. And again, the B cells are what will produce antibodies. So the B cells will go to our plasma, because remember, our plasma is what carries our antibodies, and the plasma is what will have then the antibodies that the B cells told them to produce. The dual nature, and I know this is a lot of information, guys. I'll do another kind of study guide type situation so you know kind of what specifically to study, especially from this chapter. The dual nature of the immune system. So lymphocytes, again, are kind of major class of white blood cell, give rise to two types of immune responses: antibody-mediated and cell-mediated. Antigens can trigger both types of responses. And again, an antigen is anything foreign that our body takes in. Both types are able to recognize the self versus non-self, be very specific, and have memory. So whether it's an antibody-mediated or a cell-mediated immune response, our body is able to recognize if it's a self-antigen, like on our red blood cells, or an antigen that's completely foreign. They're able to be very specific for these antigens. And then once they bind those antigens, they're able to remember that specific antigen. So they'd be able to remember a virus. You know, for example, if a virus comes into your body, here's that antigen virus would have, would go through this immune response. Your body then would remember any future antigens of the same virus. And this is where vaccines come into play. And I think we talk about vaccines at the end. So antibody-mediated immunity is effective against antigens in your body fluids. It's effective against bacteria, viruses, toxins. What antibody-mediated immunity does is once it sees an antigen, your B cells will then produce an antibody against that antigen, against that foreign substance, whether it's a bacteria, a virus, or a toxin. An antibody structure looks like a Y, if you remember a little bit from our blood discussion. The variable region is this V part of the Y, and it will bind to the epitopes of an antigen using an antigen binding site. What that just means is that this V part of the Y-shaped antibody structure is very specific for a specific type of antigen. And then the stem of the Y, which is the part just going down, is each class of an immunoglobulin that has the same structure. So here we have our antibody structure. This is the V-shaped region that's very specific for antigens to bind to. And then the constant kind of stem region going down is the site of binding to macrophages, basophils, and mast cells. The antigen binding site is the site on the antigen where the antigen binds. So up here, you see our antigen binding sites that are very specific to a specific antigen. There's a number of antigen binding sites on antibody, and there's 5 classes of immunoglobulins that are used to destroy antigens: IgG, IgM, IgA, IgE, and IgD. So they all look a little different. This is the antibody structures and the different immunoglobulins that will help to destroy those specific antigens that bind. Someone's knocking at my door, but I locked it, so we're gonna have to be okay. So IgG is the most common. It makes up about 80 to 85% in your serum. It will activate complement and increase phagocytosis, so this idea of engulfing anything foreign. IgG is an immunoglobulin that can cross the placenta during pregnancy and provide protection to the fetus. It's responsible for Rh reactions. So this is what, if we talked about the Rh factor being positive or negative, it can cause IgG can carry the anti-Rh antibody to the newborn if the woman has that Rh antibody in its system. So this can cause hemolytic disease of the newborn. Hemo has to do with blood, lytic has to do with lysis, so the splitting apart of the red blood cells in the newborn. IgM will act on the antigen binding receptor. It's responsible for transfusion reactions in the ABO blood system. So this is the antibody that we talked about, anti-A, anti-B, anti-Rh, reactions in the ABO blood system. And it's often the first antibody produced in response to an antigen. IgA is secreted into your saliva, tears, onto your mucous membrane. So this antibody protects your body surfaces, and it's found in the colostrum and milk to provide immune protection to the newborn directly after birth. IgE is the least common. It binds to mast cells and basophils. It stimulates the inflammatory response, and IgD functions as an antigen binding receptor on B cells. So 5 different types of antibodies. These are the effects of antibodies. So the effects of the antibodies, they will inactivate the antigen, because again, we're trying to get rid of this, whatever foreign substance we have, a virus, a bacteria. It will also bind antigens together, and it activates what we call a complement cascade that will initiate the release of more inflammatory chemicals, facilitate phagocytosis to eat up the antigen, whatever's foreign. So this is the idea of what an antibody does. So the antigen will bind to a specific antibody. It will inactivate the antigen. It will bind other antigens together, which activates kind of this cascade reaction of bringing more forces to try to get rid of this antigen as soon as we can. The antibody kind of binds to a mast cell or a basophil to produce an inflammatory response, and eventually that antigen will be eaten up by a macrophage via phagocytosis. Antibody production, and we're getting close to the end, so you guys are doing great. The primary response of an antibody, so how are our antibodies actually produced when an antigen, anything foreign, enters our cells? The primary response is the first exposure of the B cell to the antigen. So remember, the B cell is what will go on and create the antibody. So once the B cell is exposed to the antigen, the B cell undergoes division and forms plasma cells and memory cells. The plasma cells are what will produce the antibodies, and in about 3 to 14 days, they will be effective against that antigen. The person may develop disease symptoms, but then in 14 days, these antibodies will be completely effective against that foreign substance. And then a secondary response involves memory cells. And this occurs when your immune system is exposed to an antigen that it has seen before. Your B memory cells will quickly divide to form plasma cells, which produce antibodies, and this produces new memory cells. So it will this will happen a lot more quickly because it will understand and remember that it's seen this foreign antigen before. So this is a look at how antibodies are produced in the primary response, which occurs when a B cell is first activated by an antigen. The B cells proliferate to form plasma cells and memory cells, and the plasma cells will produce antibodies. And it usually takes about 3 to 14 days after the first exposure to create those antibodies, and they'll create much fewer. But then a secondary response occurs when another exposure. So when these B cells are exposed to the same antigen again, it causes the memory cells to form these plasma and additional cells and antibodies a lot more rapidly. And this secondary exposure then will be much faster and produce more antibodies. So that's antibody-mediated immunity. Cell-mediated immunity is used against antigens in cells and tissues, and it will be effective against any sort of bacteria inside your cells, viruses, fungi, protozoa, which are a type of living organism that should never be in your cells as well. It uses different types of T cells, helper T cells, and cytotoxic T cells. So your helper T cells activate macrophages, say, hey, we need your help. They help to form B cells, and they promote the production of these TC, which are cytotoxic T cells. The cytotoxic T cells are the precursor to cytotoxic T lymphocytes, which are toxic to cells. Cytotoxic T lymphocytes then are kind of your body's biggest kind of killer, like getting out the big guns. They destroy antigens directly on contact. You have regulatory T cells, which turn off the immune system response then when the antigen is gone, because you don't need to fight it off anymore. So here we have target cells, the cytotoxic T cells, and then the processed antigen with our MHC complex, which just again helps us to process it and understand what that antigen is. The cytotoxic T cells from interleukins, from helper T cells, the cytotoxic T cells will then go on to produce more daughter T cells. Stimulation and effects of T cells. So once we create more daughter T cells, the T cells will form cytotoxic T cells or memory cells, and the cytotoxic T cells will kill cells directly on contact. That happens basically when it attaches to a target cell, and the target cell just lyses or splits apart right away. Cytotoxic T cells can also release cytokines, which help to produce inflammation, initiate more phagocytosis, and activate more T cells. So this takes you through kind of all the immune reactions. So what happens when an antigen enters your body? So innate immunity is the general response that does not approve with subsequent exposure. So innate immunity is what we're all born with. We have physical barriers like the mucous membranes, our skin. We have chemical mediators that all interfere to try to prevent a viral infection. Then when the antigen, if adaptive immunity is then the specific response that improves with subsequent exposure, it begins with a macrophage presenting an antigen to a helper T cell. So then adaptive immunity is what you kind of gain later on in life. And this is kind of then what we talked about. We have antibody-mediated immunity and cell-mediated immunity. And basically antibody-mediated immunity was the production of B cells producing antibodies to direct effects against the antigen, and also memory cells, which are responsible for adaptive immunity and secondary response or secondary exposure to that same antigen. Cell-mediated immunity was the idea of making memory T cells for the same reason, or cytotoxic T cells, which are, again, are killing our cells directly on contact. Okay, types of adaptive immunity then. So this is where we can get into some natural acquired immunity. So naturally, we acquire immunity, which is very good to do. So we can have active or passive naturally acquired immunity. Active form is natural exposure to an antigen, which causes the production of antibodies. It can be lifelong. So for example, is if you get mononucleosis, your body will remember that, and if you won't get it again. Another example is chickenpox. You guys are probably too young, but before I was young enough where I didn't get the chickenpox vaccine. They didn't have it yet, so I got chickenpox when I was 5. So that's actively, naturally acquired immunity. So you will actively get some sort of virus, and then your body will be able to, that natural exposure to that foreign substance, that virus, will cause your body to produce antibodies, and it can be lifelong. Then passive naturally acquired immunity is the transfer of antibodies from mother to child. This can occur in the breast milk, but if women, don't or can't breastfeed, this also occurs in the womb through the placenta. So this is passively acquired immunity as well, passive naturally. And then artificially acquired immunity, this is the idea of immunization. So this is an injection of an antigen. So using a vaccine, which causes the production of antibodies. So it's bringing in something foreign, purposely putting it into your body so that your body will be able to produce antibodies against it, and then later on be able to remember it if it gets exposed to it again, the same antigen. Immunization is a process of introducing a killed live or inactivated pathogen so that your body can then go through this whole system that we talked about to produce antibodies against it. Yes, Kim, I heard that too. You would put kids together just to kind of get it over with, because chickenpox was a lot better to get when you were young versus getting chickenpox when you were older. It was a lot more severe. I wasn't purposely put with a kid, but my best friend at the time, when I was 5, he had chickenpox, and I think I just got it from him. But yeah, they would do that. And then passive artificially acquired immunity is an injection of an antibody from another person or another animal. So this is where immunizations come in. Very good. The things that we've been able to eradicate. We don't have to get into a vaccine discussion, but if you think about polio, rubella, mumps, measles, years ago, those were really scary to have, and the immunizations against them have really helped eradicate some of those from our society. So I'll just say that about immunizations. And then, so here are the ways to acquire adaptive immunity. So again, this adaptive immunity is the ability of your body to remember something foreign so that if you get exposed to it again, it can fight it off. We have natural and artificial active immunity, where the immunity is provided by the individual's own immune system. So natural is an antigen introduced through natural exposure, like chickenpox. Artificial is an antigen deliberately introduced in a vaccine, for example, but that will provide an active immunity, because that vaccine, you're kind of purposely injecting someone with some sort of exposure to a virus, but then your body, it will be able to fight it off and then produce this adaptive immunity. Then passive immunity is immunity transferred from another person or an animal. So natural usually is transferred across the placenta in breast milk. Artificial antibodies are produced by another person or an animal will be injected. So with that, I'll stop the recording, because this is probably plenty for you to take